Hello everyone. Today, in the subject of medical microbiology, we are going to study the topic named biosafety levels. The objective of this topic is to understand what is the real meaning of all biosafety levels, what BSL stands for, what are the intricate details involved in it, and what are other scenarios. So, what will be the outcome? Outcome of this topic will be that you would be able to understand fundamental applications of particular topics that are employed in working on pathogen under the containment facilities. So coming on to the biosafety. What is biosafety? First of all, we must understand the real meaning of biosafety. Biosafety is defined as the discipline addressing the safe handling and containment of infectious microorganisms and hazardous biological materials. Okay, what you have to consider and what you have to see is that whenever you are performing some sort of experimentation and when you are safe handling the highly pathogenic uh, strains and highly pathogenic microorganisms, they should be contained inside a controlled environment, inside a controlled laboratory. The practice of safe handling of pathogenic microorganisms and their toxins in biological laboratory is accomplished through the application of containment principles and risk assessment. So, whenever you are handling these pathogens, when you are handling these microorganisms along with their toxins, right? So, you are in a very compromised situation. So, you should always have laboratories that are having all the containment principles and facilities where you can work safely with those organisms. And in this image, we are showing the biohazard symbol and all the laboratory individuals who continuously work on the forefront in such a pandemic situation of COVID-19 and other variants that are in the in this world and continuously working for the betterment of the society. So we will be undergoing all these levels. Okay, in this lecture, we will be covering all the biosafety levels with regard to all the standards, with regard to all the different stages. And here you can see that it starts from BSL 1, BSL 2, BSL 3, and BSL 4. So BSL 1 is the most non-pathogenic organism where most of the time students are trained, and BSL 2 where slightly pathogenic microorganisms are taken and they can be uh, not that much dangerous. And but the real difference starts with BSL 3 facility, and where pathogens like mycobacterium, tuberculosis, and further on BSL 4 like Ebola, coronavirus, they are all worked under those laboratory conditions, under those containment facilities. So, regarding to that, first of all, you must make your concept clear about RG. Okay, the full form of RG is risk group. So it starts from risk group 1, 2, 3, and 4. Okay, so what is the meaning of RG and what it really stands for? Like RG1 is a risk assessment, no or low individual or community risk, like a microorganism that is likely to cause human or animal disease. Okay, so moreover, the real summary is that when a person or an individual or a scientist or a research scholar is working under laboratory conditions, all the microbes have been categorized under the risk group population. Okay, if he or she get infected and goes out into her his or her family, and in the main process he infects some other individuals, and what is the percentage and what are the chances that that particular infection will be a community infection or an outbreak, or uh, is the family at risk, is the community at risk? So all those organisms have been individually categorized in this fashion. So risk group 2, moderate individual risk, low community risk. So a pathogen that can cause human or animal disease but is likely to be a serious hazard to laboratory workers. It is very unlikely to be. Uh, the community, livestock, or environment. Laboratory exposure may cause serious infection, but effective treatment and preventive measures are available. And the example is Streptococcus, herpes, most of the mammalians and mice. So, whenever you are working on these microorganisms, you have to be careful whether they are pathogenic or not, whether they are dangerous or not. But you should always treat them as pathogens. Pathogens are pathogens. So risk group 3, high individual risk, low community risk. If the individual is being affected, the risk is very high. Okay, the chances of catching infection is very high. But low community risk, the community is less likely to get infection from that very individual. A pathogen that is usually serious human or animal disease but does not ordinarily spread from one individual to another. Okay, like HIV, SARS virus. Okay, it needs a lot more than a personal contact for spreading from one person to another. Like risk group 4, high individual risk and community risk. Right, a pathogen that usually causes serious human or animal disease that can be readily transmitted from one individual to another, directly or indirectly. So you don't need to take the example of that. Like COVID virus is the first hand example of risk group 4 organism or pathogen. So this is the main table, and most of the time when you are studying about biosafety, these groups are readily used in these short forms, and these terms are readily used here and there. So whenever you are studying these things, you must be well versed in the short forms and in the synonyms of these words. So here we are starting with biosafety level 1. Biosafety level 1, or also known as BSL 1. Biosafety level 1 is the level appropriate for working involving well categorized agents not known to consistently cause disease in immune competent adult humans and cause a minimal potential hazard to laboratory personnel and environment. So most of the time, the individuals who are working in these kind of laboratories that are BSL1, they are pretty safe because the agents that are being worked upon, the agents that are being used, they are not at all that much risky. Okay, so they are not known to be a real threat, or they are not known to be highly pathogenic or highly contagious in nature. Biosafety level 1 is the lowest safety level, and the precautions required for the level are thus limited and not as extensive. So, most of the time, the individuals are trained, the students are trained in these laboratories because uh, good hands on training is very much essentially required when you are in the field of medical microbiology or you are working in biotechnology. These laboratories provide a journal space in which work is done with viable agents that are not associated with disease in healthy adults. So, agents that are being worked upon in these laboratories are mostly agents like E. coli, non-pathogenic agents of E. coli are used because sometimes students they commit some mistake or they do some mistake, so it will not be at the end of the day proven to be very lethal for them. So, what are the requirements? The BSL-1 laboratories are not necessarily separated from the general traffic in the building. Okay, most of the time they are situated in college campuses or the university campuses, and they are not at all separated from the main building because most of the time these buildings they have classrooms also which are nearby and other facilities also which are nearby these laboratories. So whole soul story is that there's no separation is required for such facilities. Most of the most of the work is typically conducted on open benchtops using general microbiological practices. So the main aim is that in BSL-1 laboratories, the students they should be trained, and they are expected to be trained in the general microbiological practices. They should know the normal handling which are required in the microbiology lab, and what are the other essential things that are required, the skill set that is required. Unique laboratory design or containment equipment are not required but may be used depending on the risk assessment. So, most of the time, uh, other BSL-3 and BSL-4 laboratories are designed in a particular fashion where they install their uh, particularly specialized workbench and their facilities. But those equipments, okay, those biosafety cabinets are not the part of BSL-1. Although you want to install them or you want to work on them, you can bring them in. Okay, BSL-1 has already got the vacant space and where you can install all these facilities. But most of the time, the real scenario, the working scenario is just open bench top where the students they practice their all those skills. Okay, where they are just learning. Laboratory personnel must be provided with specific training in the procedures to be conducted in the laboratories, which is then supervised by a scientist with training in microbiology or related areas. So when the students are being trained, so whatever the high-ranking official is present there, he or she is fully or whole in charge for the training of the students in all the certain areas with all the skill sets required. The following are the standard practices, safety equipment, and facility requirements that are required in BSL-1. So, what are the standard microbiological practices? The laboratory supervisor should implement the policies regarding the access control to the laboratory. Laboratory personnel must wash their hands after working with potentially hazardous material and before leaving the laboratory. So these are the some most important hygiene practices that should be immediately implemented and immediately should be adopted by the students or the research scholars or the trainees there. So they should always wash their hands, and whenever they are touching hazardous material and before leaving the laboratory, they should always wash their hands. Activities like eating, drinking, smoking, handling contact lenses, applying cosmetics, and storing food are not to be permitted in the laboratory areas. Overall scenario is that whenever you are working in the laboratory, you should avoid the things that include personal contact. Okay, so nothing should be in your close contact. Mouth pipetting is prohibited. Mechanical pipetting devices must be employed. Okay, because you don't know before you were there, anyone else was working on that very particular pipette, and what sort of experimentation he or she might have done, what type of microorganism he or she might have used. All procedures to be conducted in the laboratory should be performed while avoiding the creation of splashes and aerosol. So here in this image, you can see that a lady is working and she is wearing gloves and she is handling the micropipette and she's very keenly doing the experimentation work and pipetting. Although she is also wearing the eye gear that is required because you don't know when you can create a splash in the chemical and any aerosol. So in that very portion, you need to require an eye gear that is essentially important. The work surface, like the bench top, should be disinfected after the work and any after any spills of potentially hazardous biological material. They should always be sterilized. Bench tops should be cleaned up properly. Whenever you finish your experimentation work, it is your own duty to leave the area clean. You should never leave the glassware dirty. You should never leave the work area dirty. The supervisor must ensure that all the laboratory personnel acquire appropriate training and necessary precautions while performing their tasks. So, uh, the supervisor must make sure that every individual who is working there should be highly trained and should be well versed in the protocols that are essentially implemented in that laboratory. Because every time when you are training someone, then you need to be effective supervisor, then you need to be effective trainer, right? So when you are readily trained, when you are work ready, then only you can give demonstration, give training to other individuals. So what are the safety practices? There are no safety specific safety practices required for BSL1. And safety equipment, special containment devices like biosafety cabinets are not required for BSL1. In order to prevent the contamination of personal clothing, protective laboratory coats, gowns, or uniforms are recommended. So all these equipment like laboratory aprons and other clothing are just meant for the personal safety and security of the individual, because during the experimentation process you don't know when you are going to spill some chemicals, when you can get a splash from an acid or any other harmful chemical. So in that whole process you are protected under that lab coat. While conducting tests with high possibility of aerosol formation, protective eyewear can be used. Wearing an eyewear is a must whenever you are handling chemicals. Uses: Biosafety Level 1 is commonly used while performing tests on microbial agents that are not known to cause diseases in immunocompromised individuals. This is the foremost important thing when you are dealing with BSL1 laboratories. For example, if any pregnant lady is there or any individual whose immune system is not up to the mark, they all can come and go from the laboratory. There's no risk involved. These laboratories include the laboratories used for teaching purposes in colleges and training centers. The main purpose is to give certain individuals good hands-on training of basic microbiological techniques and biotechnological techniques that are involved all over the place. So organisms, as we all know, that organisms that are not at all pathogenic in nature, like Pseudomonas, E. coli, Lactobacillus, all those types of containment, they don't need some sort of containment. However, the requirement of biosafety level might differ depending on the risk assessment of the pathogen. But most of the time experimentation is performed on the uh, microbes that are not at all pathogenic, because trainees might sometimes falter or they might commit some sort of mistake. Now, moving on to the biosafety level 2. Biosafety level 2 laboratories are the laboratories that are used for the task involving microbial agents of moderate potential hazards to the laboratory personnel. So they involve microbial agents with moderate hazard. Okay, the environment and the agent. However, the infectious agent or the toxin might pose a moderate danger, not a high risk of danger, moderate danger, if accidentally inhaled, swallowed, or exposed to the skin. Most of the time, when you are working in these facilities, the maximum level of infection that occurs, or the what you say accidental exposure, is due to inhaling only. The precautions associated with biosafety level 2 are comparatively more extensive than BSL-1, but BSL-1 and BSL-2 laboratories are generally considered as basic laboratories. This is true because every time when you are training each and every individual in basic medical microbiological technique, you need a slight variation. Okay, level 1, now you move on to the level 2. Before moving to level 3 and 4, because when you move towards level 3 and level 4, you are already expected to be well versed in all the technologies and all the techniques on all the equipments that are employed and utilized in the laboratories. What are the requirements? BSL2 laboratories like BSL1 laboratory are not necessarily separated from the general traffic pattern in the building. So they are not at all separated from the general traffic pattern in the building. They are also present in the main building or near to the classrooms or near to the staff rooms. However, access into the laboratory is limited while BSL2 experiments are in progress. So most of the time when the experimentation process is under progress, the facility or the laboratory doors are kept closed. The annual inspection of the laboratory is also an important part of the BSL2 requirement. So, most importantly, whenever the equipments are being used, there are different instruments are being used. Every equipment should be up to the mark. So, there should always be an annual inspection where everything has to be tested and annual inspection, annual audit is conducted. These might include changing filters or replacement of some devices. That's very well understood because all the machineries that are involved, they sometimes do need servicing or some sort of replacement. Because after a while or after a very long duration of use, they do suffer some sort of faults or wear and tear. The work is mostly conducted on sterilized benchtops, ex- except for some processes that might form aerosol. The latter is conducted in safety cabinets. So, safety cabinets are also present there, and somewhat it is much more advanced in comparison to BSL1 facility. The work is mostly conducted on the sterilized benchtops. The precautions to be followed in BSL2 include all the precautions of the BSL1 and some additional precautions. So it is just promotion in the level of training that is required in the field of medical microbiology and biosafety. Standard microbiological practices. All the laboratory personnel must wash their hands after using viable microorganisms. This is a must and it should be followed every time and before leaving the laboratory. Eating, drinking, smoking, handling contact lens in the laboratory is strictly prohibited. Mechanical pipetting should be done instead of mouth pipetting. All contaminated cultures, glassware, plasticware, and biologically contaminated waste must be treated as biohazard and thus autoclaved. So it is a very healthy practice whenever you have finished or completed your experimentation process, you are most likely uh, to know this thing. Okay, whenever you are discarding anything, when you are washing anything after the crucial experimentation process, you are always supposed to autoclave all the glasswares and all the things that have been employed in that very particular experimentation. And after autoclaving, you can proceed further, whether it's discarding of that material, discarding of the media, or discarding of anything because before autoclaving you are not supposed to discard it into the waste. So here in this image you can see how clean and how neat is the work area, right? So work surfaces must be decontaminated with disinfectant at the end of the day after any spills or splashes. This is the very hardcore practice that you should always understand. Whenever you have finished your experimentation process, you should always clean and disinfect your work area every time. Use hypodermic syringes, needles, plastic pipettes, razor blades, contaminated broken glass, and blood vials are treated as medical waste and discarded in a puncture resistant, sharp, disposable container. So every time you should be really careful whenever you are discarding the waste. So safety practices, people with increased risk of acquiring infections like Immunocompromised and pregnant individuals should not be allowed to enter BSL laboratories. This is the main thing you must understand while laboratories are at work. So these type of individuals are not at all welcome when the experimentation procedures are going on. An annual review of the BSL2 manual should be done to update the guidelines every time annually. Documented policies and procedures should be established that limit the entrance to the individual who know of the potential hazards and are appropriately trained every time. High level of training, high level of precaution and safety levels. A biohazard symbol is placed on the piece of equipment where biohazardous materials are used or stored, or any even the experimentation procedures are going on. On the laboratory door, the symbol of biohazards should be placed, and safety equipment. Protective coats are to be worn while entering the laboratory and then removed and kept in the laboratory post-work. The laboratory design should be made such that it can easily be cleaned and decontaminated with minimum nooks and corners. So whenever these type of laboratories are cleaned, whenever uh, they are being sterilized or decontaminated, while the construction is going on, the corners are always made smooth because the cleaning process should be very easy. The laboratory design should be like that it should be very easy to clean. It should not be problematic space to be cleaned. The laboratory doors should be closed whenever work with hazardous biomaterial is conducted. Whenever the experimentation procedures are going on, you should always close the laboratory doors. An autoclave must be available. It is a must. Joseph, biosafety level 2 laboratories are mostly used for routine analysis and culture of moderately hazardous agents. Besides, some of the laboratory used for teaching and training purpose are also BSL-2 laboratories. Coming on to the organisms that are required in BSL-2 laboratories include pathogenic strains of E. coli, Staphylococcus, Salmonella, Plasmodium, Toxoplasma, herpes, and other viruses. The allocation of organism in the laboratory, however, might differ depending on the risk assessment. So every individual has a different working scenario, and the basic design of BSL2 laboratories are very similar. There might be some differences in the installation or in the placement of the equipment. Now, coming on to the BSL3 laboratory facilities or biosafety level 3. BSL level 3 is the level where work is performed with agents that may cause severe or potentially lethal disease. This point you should take into your consideration that here the pathogens that are being used are very uh, lethal in nature and potentially severe in nature. Okay, though inhalation of aerosol formation to the personnel and may even contaminate the environment. So that particular pathogenic agent is not only dangerous for the individual but also dangerous for the environment, right? The tasks performed in BSL-3 laboratories involve indigenous or exotic agents where the potential for infection by aerosol is high and the disease may have lethal consequences. You must understand what is the real meaning of the term indigenous and exotic. Indigenous means native. Okay, any disease agent or disease pathogen that is formed natively in that very country. And exotic means any disease agent that is foreign in nature. Auto-inoculation and ingestion present primarily hazardous to personnel working with these agents at this level. Working in such laboratories require laboratory personnel with specific training in handling pathogenic and potentially lethal agents, along with supervisors, scientists competent in handling infectious agents and associated procedures. So BSL-3 somewhat is uh, very much advanced and somewhat complex in comparison to BSL-1 and BSL-2 facilities, and advisors and supervisors are expected to be highly trained individuals, those who supervise other individuals and supervise other students or research scholars. So, and they are very highly competent in handling dangerous or infectious agents. So, what are the requirements? Biosafety Level 3 containment laboratories for animal and research are the most challenging containment level facilities to design and operate. They are the most challenging ones. These laboratories should be certified for the use before initial operation and subsequently on an annual schedule after a program change, renovation, or replacement of system components that may affect the operating environment of laboratory. So after a certain duration of time, everything has to be examined, and everything has to be renovated, replaced, and serviced. Because most of the time, uh, annually, these laboratories are audited. These laboratories are inspected that everything is functioning up to the mark. Are there any drawbacks? Are the equipment functioning very well? BSL-3 laboratories are also called the containment laboratories. Containment means to stop something within the laboratory, any disease pathogen that is very contagious in nature, as they require containment equipment to protect the personnel, the microbial agent, and the environment. The requirement for BSL-3 include all requirements of BSL-1 and BSL-2 laboratories, along with some additional design features and special equipment. So design is very much similar to the BSL-1 and BSL-2 facilities, but there are some more additions in the special equipment category. Standard microbiological practices. The entry to the BSL-3 laboratory is limited to individuals with appropriate training in handling BSL-3 organisms, all of whom are selected by laboratory supervisors. We must make sure that all the individuals who are working under DSL-3 facilities are highly trained, and they all know the pros and cons of working in that facility, and they should know what will be the contagious results and the dangerous results if they mishandle any pathogen in the laboratory. Besides the general procedures and laboratory practices, the supervisor also formulates additional policies to limit the entry to the laboratory. Okay, entry to such kind of laboratories is very limited. All the procedures to be conducted in BSL-3 must be conducted within the biosafety cabinet to prevent the exposure of aerosol to the laboratory personnel. So biosafety cabinets are always installed, and they are always there, and it is ensured that you do not work outside those cabinets in order to ensure the safety of other laboratory personnel. Personnel working in the laboratory must wear personal protective equipment before entering the laboratory and then remove them out before leaving. So PPE kit is essential and the last line of defense when the individual is working in such kind of conditions. The work surfaces and sinks should be decontaminated once every work shift or after any spills or splashes. It is also done after the completion of experimentation. Whenever you are working there and you complete your task, you are always supposed to clean and disinfect the work area. Okay, it should always stay clean. The BSL-3 laboratories should be separated from the general traffic in a building to limit the entry of laboratories into the laboratories all the time. So most of the time, these type of facilities are situated in a very isolated area, away from general population, away from all kind of facilities, because minimum intrusion is required in safe functionality of these laboratories. Safety practices: the doors of BSL-3 laboratories are closed at all times with appropriate BSL-3 signs outside the suit, along with universal biohazard sign and emergency contact information. Laboratory personnel must have medical surveillance and offered appropriate immunization for agents handled or potentially present in the laboratory. So all the laboratory personnel who are working on those contagious pathogens, they should also receive uh, a very good level of vaccination, immunization, and medical surveillance because they are performing dangerous experimentations on those pathogens. Each institution should consider the collection and storage of serum samples at risk persons. A laboratory-specific biosafety manual, which is available and accessible to all, must be prepared and adopted as a policy. So every time, if new advancements are taking place, anything new or modernization is taking place, you must ensure to adopt that into the policy, and biosafety manual should be upgraded after a particular duration of time. Laboratory supervisor must check the demonstration of proficiency in the standards. Standards should never be compromised in BSL-3 facility, and special microbiological practices by all laboratory personnel before working in BSL-3 agents. Potentially hazardous materials must be placed in a durable leak-proof container or a vial during collection, processing, storage, or transport within the facility. So, when you are dealing with hazardous material, they should be always stored in a leak-proof container. Okay. All the laboratory equipment should be routinely decontaminated after work or after any spills or splashes. The laboratory biosafety manual must define procedures adopted in the case of exposure to infectious material, and these should be treated accordingly. So, if any accident occurred in the laboratory, what are the necessary steps to be taken and what are the precautionary measures to be considered all the time? No work in the BSL-3 laboratory should be conducted on an open bench or an open vessel. Everything is under containment conditions. Okay, safety cabinets are there for working. All activities involving infectious agents must be conducted within the biosafety cabinets or other physical containment devices. That's what I was explaining earlier. Safety equipment. Biosafety cabinets are used for the manipulation of all infectious agents. Individual protection gears like personal protective equipment (PPE), kit, coats, gloves, and respiratory protection should be worn while entering the laboratories and then removed before leaving. So every time they should be sterilized and disinfected before leaving and after leaving the laboratory. The airflow in the laboratory should not be recirculated to an area of the laboratory and should be HEPA filtered prior to being discharged to the outside. The filter manuals, equipments, vacuum pipes, autoclaves, etc. should be revised and renewed annually. Okay, there's no room for error. There's no room for procrastination or any careless activity. Everything should be of top notch. Everything should be flawless. Everything should be free of error. Uses. PSL-3 laboratories are used for clinical, diagnostic, teaching, research, or production facilities. These laboratories are used for handling and manipulation of highly infectious agents that cause a direct, severe effects on the health of the personnel. These are used for the studies regarding the effect of infectious agents and various toxins and their effects. So organisms that are used in BSL-3 facilities are like HIV, Mycobacterium tuberculosis, SARS, rabies virus, West Nile virus, etc. Placement of organism in different biosafety levels, however, might differ and should be determined after the risk assessment. Okay, the risky the organism, it should be placed accordingly. Now coming on to the biosafety level 4. So Biosafety Level 4 or BSL-4. In this image, you can see the common laboratory setup and the biosafety cabinet where all the individuals they work on. Biosafety level 4 is the highest level of that is employed while working with dangerous infectious agents that present a high individual as well as environmental risk in the form of life-threatening disease, aerosol transmission, or unknown risk of transmission. The BSL 4 laboratories are often used while handling and manipulating risk group 4 pathogens like COVID virus, not known vaccine or therapies, because earlier there was nothing to be that was our scientific community was aware of it. Okay, nobody knew what are its pathogenic abilities and what are the routes of infection and how it should be treated. Nothing was known, and still the scientists were working 24/7 effectively, and they developed a vaccine. So here in this image, you can see the scientists wearing biosuits along with the PPA kit inside it, and these cables are connected for providing them their individual atmosphere. BSL-4 laboratories are of two types: cabinet laboratories where all the work is performed in a Class 3 biosafety cabinet or similar physical containment with very carefully formulated precautions, and to laboratory, where all laboratory personnel are required to wear full body air supplied suits, protective gears in the form of PPE. So these body suits they provide them protection, but also inside it they are wearing PPE kits for full body protection. So what are the requirements? The requirements of BSL-4 laboratories are extensive with specific laboratory design, training procedures, and highly protective equipment and personal gears. So these laboratories should be certified before use, before initial operation, and subsequently on annual schedule. After a program change, renovation, or replacement of system components that may affect the operating environment of the laboratory. And these laboratories are also annually analyzed and annually audited for any sort of change in technology, any renovation, or any repairing. BSL-4 laboratories are also termed as maximum containment laboratories as they have secondary barriers to prevent hazardous material from escaping into the environment. Okay, because the experimentation that is going on in BSL-4 facility is of top-notch level. Okay, the most highly contagious organisms or the pathogens are being examined or being studied. BSL-4 laboratories should follow the requirement of BSL-1, BSL-2, and BSL-3, along with additional specific precautions. So, standard microbiological practices that are implemented in BSL-4. No work conducted within the BSL-4 should be done on an open bench or an open vessel. The workstations, equipment, and sinks should be sterilized post-work. Every time, the laboratory personnel should be in protective gear that might include full body PPE, gloves, masks, and coats. So, full protective measures should be avoided, always taken, and full body protection measures should always be adopted. The doors of the laboratory should be closed at all times, with the laboratory placed away from general traffic in the building, mostly situated in the isolated areas. Activities like drinking, eating, mouth pipetting should be avoided at all costs. Only people that are trained in handling the BSL-4 organisms that are of very high value, and these individuals are highly trained and highly uh, valued individuals, and the equipment in the laboratory should be allowed into the laboratory. What are the safety practices? Viable or intact biological materials to be removed from Class 3 cabinets in a BSL-4 are transferred in a non-breakable sealed primary container with a non-breakable sealed secondary container. No material except the biological material that are to remain in a viable or intact state are removed from BSL-4 laboratory, unless they have been autoclaved or decontaminated before they leave the facility. No intact pathogenic agent should leave BSL-4 facility. Only individuals whose presence in the facility is required for microbiological processes or support uh, purposes are authorized to enter. Individuals that are at risk of acquiring an infection or for whom infection may be usually hazardous are not allowed in the laboratory. Personnel can enter and leave the facility only after the clothing change and through the shower. So here you can see the image of BSL-4 facility, how the individuals are connected and how they are covered in full body suits along with uh, their PPE kit. The person can enter and leave the facility only after clothing change. When BSL-4 laboratory is at work, or when infectious material or infectious animals are present in the laboratory, a hazardous warning sign along with universal biohazard symbol is placed on the all access door. Every time everyone should be aware of what kinds of dangerous experimentations are going on. Our system is set up uh, for reporting laboratory accidents, exposures, and medical surveillance of potential laboratory associated illness. So safety equipment. A Class 3 biological safety cabinet or Class 1 or Class 2 biological safety cabinet used in conjunction with one-piece personnel suit ventilated by a life support system are to be present in BSL-4 while conducting all procedures within the facility. So it depends on the use of biosafety cabinets, how many individuals can work all together. Right? Walls, floors, ceilings of the laboratory must be sealed to internal shell. This facility fumigation and is animal and insect proof. You can see the flooring here in the image and the walls. They all are having galvanized iron and steel walls, and floors are also sealed. A double door autoclave is placed for decontaminating material passing out of the facility. Here in the image, you can see the life support system connected to the biosuits and all the equipments that are eventually placed in the use of BSL-4 laboratories. The exhaust air from the facility is filtered through the HEPA filters before being discharged to the outside so as to prevent its entry into the occupied buildings and air intakes. The uses of this BSL-4 laboratory is that used for diagnostic and research work on easily transmitted pathogens causing fatal diseases. These laboratories are used for new and unknown pathogenic microbes, for which no vaccine or therapies are available. They are also used for clinical and production facilities that require highly sophisticated techniques and advanced processes. So organisms that are used are like Ebola, COVID virus, okay, encephalitis virus, all those things that are of highly contagious nature, SARS virus. So here's a quick summary of all the biosafety levels and containment and for what purposes these laboratory facilities are used and what type of laboratory practices that are used and safety equipment that are need to be installed in all those facilities. Biosafety, basic biosafety level 1, basic biosafety level 2, containment biosafety level 3, maximum containment biosafety level 4. So this is how we finish this lecture. Thank you for listening to me patiently. Thank you so much.